Well, I'm honored, very honored to introduce Professor Mack, my brilliant colleague from life science. Today he's going to tell us about this year's Nobel Prize winning Yoshinori Oshumi, whose work on autophagy won him the Nobel Prize. And I shouldn't take up Professor Mack's time, but Professor Mack is Associate Professor in Life Science. He got his degree, all his degrees, from Cambridge University. And we are really happy that he's going to tell us why Professor Yoshinori Oshumi deserves the Nobel Prize.
Okay, so thanks to Henry for giving me the opportunity to revise autophagy for myself so that it will be useful for my own research at some point. And so since this is supposed to be for a general audience, so I'm not really going to talk about very specific kind of experiments, specific kind of details of publications. So what I'm going to do, in fact, is just to introduce you to this year's Nobel Prize winner as a person, and also kind of go through some of his landmark discoveries that he made that won him the Nobel Prize. This set of experiments are actually done very, very recently. This was actually work done in the 1990s. And it's actually to the testament of kind of the strength of this set of experiments and the universal significance of this set of experiments that he won the Nobel Prize so quickly. And obviously, the other remarkable thing is that even though the maximum number of people can win a Nobel Prize would be 3 per year, this year the Nobel Prize for Physiology and Medicine is actually won by a single person, which is what Professor Tai said, by Yoshinori Oshumi, who is currently a professor at the Technical University of Tokyo, which we'll see later. So Professor Oshumi was cited for his groundbreaking discoveries of the mechanisms of autophagy. So some of you may have heard this term being pronounced as autophagy. I'm going to say autophagy. I think this is kind of reflection of where you came from, where you learned this particular word. And I really like this picture because I think for any bench scientists, they're always most at ease in the laboratory. And I hope you can kind of sense by the look of Professor Oshumi here. Okay, so just a quick background on the personal history of Oshumi. So he was born in Japan. He received his PhD from the University of Tokyo. He went abroad to the United States at the Rockefeller University for postdoctoral work for 3 years before returning to Tokyo as a research associate. So he didn't actually immediately establish his independent research group. In fact, it took him many, many years before he actually managed to secure his own funding and his own independent position. So up to 1988, he was still under the umbrella of a more senior faculty member at the University of Tokyo. And so in 1988, he finally achieved independence. And he assumed the associate professor position at the University of Tokyo. This is the first time he established his independent research group. From an interview of him, actually, he said that he was, by then, in 1988, he was 43 years old, and he was very honest in saying that he was not very successful up to that point. And he had many difficulties. And I think this actually says a lot about kind of in science, you don't always become successful instantly. I think it takes time sometimes for you to really study your questions and ask deep questions that may not actually give you answers immediately. But since the establishment of his first research group, he became very successful because he actually chose the right question. And so he then moved to National Institute for Basic Biology in Japan up till his formal retirement age in 2009. And then afterward, he moved to the Tokyo Institute of Technology, as the professor since 2009, where he's currently resided and where he's still running an active research program. So, and this is actually also kind of very telling in kind of understanding the philosophy of the way he thinks about scientific questions and how to choose directions of research. So he was asked on his motivation behind studying the vacuole, which you'll hear a lot more about later on, and autophagy. And instead of someone who may have told you that, well, he has divine intervention or he has the insight or the foresight into thinking that this is a particularly important question, he instead said the vacuole was thought to be just a garbage can in a cell, and not very many people were interested in its physiology. And so he thought it would be good to study transport into the vacuole because he would not have much competition. Very practical, yet it was a great choice. And that also kind of reflected his personality, because he said he was not very competitive, so he always looked for a new subject to study, even if it is not so popular. But it turned out that he really have, by staying away from the limelight, staying away from a popular scientific question, he actually chose something that turned out to be very, very significant. And in the Nobel Prize announcement this year, 4 of his works, publications were cited. These were the 4 key publications among many that his lab published. And I think it was very significant in the sense that when you look at this list of publications, the very first 2 publications were actually published in what we normally would be calling not so glamorous scientific fashion magazines. These 2 seminal papers and really kind of laid the foundation for all subsequent work in the molecular study and mechanistic studies of autophagy was actually published in Journal of Cell Biology and in a journal called Fab's Letters. But these are the 2 papers that were actually referred to hundreds and thousands of times, and this was really the basis of all the work that not only was developed in Nosumi's lab, but also around the world. And then he actually worked on individual proteins and genes that he identified in these initial work and provided biochemical, mechanistic insights into how some of these proteins orchestrate the autophagy pathway and the autophagy process. Now, so today I'm going to break this particular presentation into a few parts. Actually, we'll go back in time a little bit more before Osumi actually even got started in autophagy, and telling you a bit more about kind of the destination of autophagy, or the final destination of the autophagy process, on the brief history of lysosome or vacuole. Vacuole is a name for lysosomes, but specifically for budding yeast. And then I'm going to touch on the pioneering work by Osumi on the few pieces of work that were cited in the Nobel Prize announcement. We'll consider kind of why autophagy is important. So this is kind of drawn from the title of today's presentation. So why recycling is good. And we'll finally consider what happened when recycling went wrong, when there's no recycling. And so this will touch upon kind of the significance of autophagy, both in health and in disease in humans. So autophagy was actually a phenomenon, and that's actually a process that was not named by Osumi, but actually named by another Nobel Prize winner. And this is Christian de Duff, who discovered another organelle called lysosome in the cell. So Christian de Duff was a Nobel Prize winner in 1974 with Albert Claude and George Palade for their discoveries concerning the structural and functional organization of the cell. This essentially means that in eukaryotic cells, in cells in our body, the cells are not only surrounded by a single envelope, a single bag. Right? Inside a bag, there are actually additional compartments Some of that may be small, some of them large, of different shape and sizes. And Christian de Duve, Palladio, and Claude were the pioneers in describing the structure of what's inside a cell in detail using biochemistry, microscopy, and lots of kind of new techniques that were important and applicable later for all other people who study cell biology. So Christian de Duve identified lysosomes, but actually his original interest was not in lysosomes, or his original goal, his scientific goal, was not to identify these new organelles, these new bags inside a cell. He was actually trained as a biochemist. He was interested in actually maybe what you would call a more medical and physiologically important question. He was actually interested in how insulin regulates blood sugar. His idea was that he wants to look for a particular fraction of a cell or a particular location inside a cell where enzymes would be located that are specifically designed or specifically responsible for processing sugar. So he was actually very successful in doing this. The technique he used was a technique called differential centrifugation, essentially meaning that you take a bunch of cells, you want to break the cells into, you want to break basically the large shopping bag that you have, even though inside your large shopping bag you still have vegetables, you have your fish, you have all sorts of other things that are kept in your free clear plastic bags these days that you'll get from supermarket. But he broke the outside bag, and because each of these clear plastic bags are now containing different contents, some of them are heavier, some of them are lighter. So by actually just spinning them at different speed, because of differences in density, differences in weight and density, these different contents would come down to the bottom of the tube. And so by doing this so-called differential centrifugation, he could separate the cell into a fraction that contains primarily the nucleus, where DNA is normally found. He can find mitochondria, which is the powerhouse where energy is mostly generated in a cell, in a mitochondria, in another fraction. And he also found another fraction that he called microsomal fractions, which turn out to be the endoplasmic reticulum in a cell. And when he asked the question then, once he was able to separate the cell into these different fractions, he asked the question, where could he find the enzymatic activity that was responsible for processing sugar? He was able to find that in the microsomal fractions, that was the place where the enzymes for processing sugar was located. That was all fine and good. I mean, he actually reached his goal. But he also had an unexpected observation in the process. In the process of finding the enzymes that he was looking for, he also noticed that there was actually another enzymatic activity in the same fraction of the cells, in the same plastic bag. So in this particular plastic bag, it's not only holding vegetables, apparently, it's holding something else. He didn't know what they were, but they were behaving distinctly different from the enzymes that was processing sugar. What he found, in fact, was that this enzymatic activity was only active after the membrane preparation was left to rot, essentially, in the fridge for 5 days. So sometimes, if you have leftover food, they taste better. They marinate in the fridge for 5 days, and now you come up with a different kind of food. And this particular flavor, this enzymatic activity, in Christian de Duve's mind, he likened this to be, or he kind of maybe put it in a more scientific way, in saying that perhaps this enzyme was trapped in a compartment that prevented it from getting out and a substrate from getting in. So essentially, there are actually even smaller plastic bags in the larger plastic bag that he found, the sugar processing enzymes. And this smaller plastic bag that contained this dormant enzyme in fact, turned out to be, the smaller plastic bag turned out to be the new organelle that he called lysosome. So he named this new organelle lysosome because this is a Greek name for digestive body. So this is actually an organelle within the cell where things are broken down into small, tiny pieces. This is the home, it turned out, for enzymes that favor a particular neighborhood, a particular environment. And this environment usually have very low pH, meaning that they like to work in acidic environment, like they like to work in vinegar, perhaps. And this is just a picture from his original paper that described the discovery of lysosomes, showing the purity of his preparation. And all these are dark lysosomal bodies where a collection of enzymes that like to live in low pH would be found. Okay, so that was 1950s. Ever since the discovery of lysosomes, lots of other people became interested. But at the time, it was also kind of thought that lysosomes was involved in digestion of both extracellular material, so things that come from outside of the cell, or of the intracellular material that are segregated by autophagy. This was the abstract from a meeting in the 1960s, and this is actually a seminal meeting because this particular meeting saw the birth of many terms that actually, as biology students, would learn in textbooks these days that include this word, autophagy, which is kind of the key for today. So autophagy was actually a term invented by Christian de Durf, thus describing the process of the delivery of intracellular material to the lysosomes. So what are the examples of autophagy? So autophagy happens in us right now as you're listening to me. And one of the very important process that involve autophagy is actually the elimination of mitochondria from mature red blood cells. So many of you would know that in red blood cells, these are the blood cells that are dedicated to carry oxygen around our body. And in red blood cells, there are actually not much of other organelles. This is really just a bag or hemoglobin, which is the oxygen-carrying chemical. There's not much else. But they started off actually with everything that a cell would have. But during the process of maturation, during the process of becoming a red blood cell, they actually threw away the nucleus and threw away other things inside a cell, including mitochondria. And it turned out that elimination of mitochondria from red blood cell precursors, as they become mature red blood cells, actually requires autophagy. Okay? So autophagy is self-eating. Or you can think of autophagy as a process of recycling. As the weather turned cold, you may have started taking out your fleece jacket for today or for the next few days. I wonder how many of you actually know that actually a fleece jacket could be made out of plastic bottles. And this would actually be kind of a practical example where recycling is good. And actually during the process of recycling, you take one form of material, or you take one product, and you can actually break it down into its basic components, turn it into raw materials that can actually be now used to make something completely different. You can also make the same thing. You can actually take plastic bottles and remake plastic bottles, but also you can also use it to make something else. The important part is that actually you need to break them down before you kind of build it back up again to form something else. So, and this is essentially what happened for recycling inside our cells, and that involve autophagy and lysosomes. Because inside a cell, basically we are composed, each cell is composed of proteins, nucleic acid, including DNA, and fat. When we have too much of these, or when some of them are no longer needed, or if they want to make something new, but out of the basic building blocks that were originally made to build these existing products, then they have to break them down into lysosomes by the process of autophagy into these building blocks. And they serve both as raw material for new structures in a cell and also for energy. Because another time when actually you need autophagy would be the time when you're starving. When you have no food, you need more food. You need something to burn, just like if you're cold, I guess in old days, you want to get wood to burn. And so you chop the tree down to get wood. And so this will be same idea that you chop something up and to give energy in this case. But the key question then becomes, how did intracellular material, how did these precursor forms of biological material reach the lysosomes? Right? And so that's actually the key question that was answered by Yoshinori Osumi's work on autophagy. So in the next section, I'm going to go through some of the key discoveries made by Osumi that clearly demonstrated, first of all, the occurrence of autophagy in budding yeast, which was his favorite model organism for use to study biology, and how he then managed to use budding yeast as a model to discover genes that are important for autophagy. And so the very first paper in his work is in 1992. This is when he first demonstrated that autophagy could happen in budding yeast, and that this is actually a readily observable process after using some genetic tricks to tell the yeast to do what he wanted them to do. So just to clarify, so budding yeast, the lysosomes, which Christian de Durf discovered originally in liver cells in mice and rats, in the yeast, lysosomes are called vacuoles. So these two names are interchangeable. I mean, these are equivalent organelles. And Osumi was interested in how vacuoles function. What he did was, and he also wanted to actually observe the process of autophagy. He actually had great difficulty in looking at autophagy initially, because actually autophagy is a process that was very efficient. And so he could actually never saw cargoes or actual material getting delivered to the vacuoles. Because once they got delivered, they got broken down into tiny, tiny pieces, and therefore they were no longer detectable using conventional microscopy techniques. So he resorted to a genetic trick in the sense that he tried to block the process of recycling inside the vacuoles. He stopped the machines from working in the recycling factory, and therefore, once these materials are delivered to the lysosomes or to the vacuoles, they are stuck in the vacuoles. And only then, he was actually then able to see that there was this gradual accumulation of material inside the vacuoles in yeast as the yeast is starved for the time of a few hours. And so in his seminal experiment, he showed that at the beginning of the starvation time course experiment, the vacuoles appeared as a clear sac, as a clear compartment. Nothing was inside. Perhaps it's because yeast was actually eating. They were getting food from outside, and therefore there's no need for recycling at that point. However, if you now stop feeding them, essentially, then they have to start looking inside the cell, looking for materials to get energy. So, and over the course of 3 hours, and by blocking the recycling machines inside the vacuoles, Osumi could actually now see the accumulation of cellular materials getting delivered to the vacuoles and stuck there. And so he basically have kind of froze this process in time in order to be able to actually observe these materials getting delivered. There was light microscopy. He subsequently also used electron microscopy to observe the same process. And by using electron microscopy, he was actually able to zoom in to these so-called autophagic bodies or autophagosomes, these are the smaller plastic bags that were getting dumped into the vacuoles. And by just looking at the morphology, he could actually see clearly where these material originated. In this things, for example, in this particular panel, he was able to infer from the morphology or the look of this particular autophagosome that they were originally coming from the endoplasmic reticulum, because there are ribosomes, these dark, small dots that were only enriched in endoplasmic reticulum in a cell. So this was key because he was now able to show that using a much simplified system, that autophagy process is conserved, and also because the yeast has so many well-developed tricks for doing molecular genetics, he was able to then use yeast as a system to now identify genes and proteins that are important for autophagy process. And so the summary of the observation of his initial work was that there is, and at that point it was a model, that there was actually a dedicated membrane system that will progressively enlarge and trapped redundant or damaged material inside the cell and to mature and form this so-called autophagosome. And the autophagosome would be the ultimate kind of delivery van to go to deliver these material to the lysosome, where the materials are broken down into tiny pieces. And then Osumi wanted to ask, what are the proteins that are important for the maturation of autophagosomes and for the delivery process? So again, using yeast, he did something that yeast geneticists would love to do, or any kind of geneticists would love to do. He wanted to then ask for mutant yeast that fail in this process. And in doing so, he would be able to actually identify proteins that are required for the process. And so the second very important piece of work was the genetic screen for autophagy mutants in yeast. And this is the demonstration of the very first mutant that was defective in autophagy. He called it ATG1-1. And you have seen this before, the lower three panels, where upon starvation, the yeast would accumulate these cargoes that was bound for recycling. However, in his autophagy mutant, the delivery process was broken. And so there's no longer anything inside a vacuole, even though this set of yeast was starved the same way as the yeast in the lower three panels. So this indicates that this particular mutant was actually failing to deliver the cargoes that are necessary for recycling. In this screen, he was very successful in identifying not just one mutant, but actually 15 different yeast mutant strains that were unable to do autophagy in various kinds of ways. And this is actually purely done by using a nasty chemical that damaged the genome of the DNA of the gene of the yeast, and looking for any sort of defects in autophagy. He named these genes from ATG1 to ATG15 for autophagy related. And what was gratifying for him, and I guess for the rest of the field, was that over the years, he was able to demonstrate, also together with other groups around the world, that the initial 15 mutants and many more beyond the initial 15 genes that were discovered in yeast actually found the counterparts in mammals and in us. Right? So actually, there's not so different between this and that, except that if you now look at this list, in mammalian systems, some of the yeast proteins is only represented by one single representative, but in mammals, there might be more than one members of the same family. So we're similar to yeast, but not quite. But this is important because actually, the demonstration that autophagic process is conserved and that the genes that are required for autophagy in yeast is also just as important for autophagy in mammals and in humans actually sparked the interest in a lot more research groups in the question of understanding autophagy. So, and in the late 1990s, he went on to clone these 15 genes that he discovered initially from his genetic screen, and actually using biochemistry to understand the function of the proteins that are encoded by these genes. And so he cloned them, and one of the two key findings of these functional characterization or these functional biochemical studies was that he found a protein complex that acted very early on in the autophagosome formation. So the precursor of autophagosome is called a phagophore. And so this is basically a patch of membrane inside a cell where proteins are progressively being attracted. But how did these proteins know where to go? Part of the important process is actually the initial recruitment of this protein complex called ATG5 and ATG12. The other stunning thing about this particular study is that he also demonstrated that these two proteins, the way they interact is actually by physical covalent linkage. So they were actually completely attached to each other. And the attachment is irreversible, which is actually quite uncommon in many of the biological protein complexes that normal people would study. Okay, so, and next, he was able to show that this ATG5, ATG12 complex was important to subsequently recruit yet another protein that he found from his mutant collections called ATG8. And in this case, he demonstrated yet another rather unexpected biochemistry, a biochemical process in a cell, in that this protein is actually covalently attached to a lipid. And this is actually the way that this protein stays on the membrane of this ever-growing autophagosome. ATG8, or LC3 in humans, actually attracted a lot of attention because this was the first marker that allowed not only researchers using yeast, but also researchers using mammalian cells to observe the autophagic process that happened in real time. Because this is such an important protein that needs to attach itself to the autophagosome membrane. Okay, so this is to demonstrate the use of LC3. So as you watch this movie, this is a time-lapse experiment. LC3 is labeled in green with the green fluorescent protein. And the cells have just been taken off food, so it's getting more and more starved. And so you're observing the autophagy process as it develops over time. I hope you would appreciate that there's actually more LC3, and they are recruited progressively to the autophagosome membrane upon starvation. And the LC3 binds the autophagic cargoes, and actually that's the reason why LC3 is so important, because it is the adapter that link the cargoes to the autophagosome. And so over time, I hope you would see that once, when you stop listening to me, then you could see that actually there's more LC3 GFP, and that there are more and more of them, and the particles or the bags that contain GFP LC3 is getting much larger over time. Okay, so I have given you one example previously why autophagy is important in the development of mature red blood cells in getting rid of mitochondria. So the other important function of autophagy and why autophagy is such an essential process is because this is a way we can deal with starvation at a cellular level and at an organismal level as a whole animal. And also it's actually led to additional mechanistic insights into what triggers autophagy. And so this is recycling for good causes, and particularly how autophagy is able to respond to starvation. And so when you're eating delicious ramen noodles in this case, autophagy does not work because you have plenty of food, your cells are happy, you have a lot of energy. The reason is because the ramen noodles, the energy from the ramen noodles is translated into signals from a protein complex called mTOR. The name is not important here, well, at least not for this context. And so at times, when there's plenty of food, the autophagy process is suppressed. You don't need it. There's no need for recycling because you have enough materials coming from outside. However, once you're done with ramen noodles, or once you're traveling and you have no access to food, autophagy kicks in to provide the energy to fill the gap of the missing energy that your body still needs. So two processes happened. One is the inhibition of the mTOR pathway, and second is the activation of a second pathway anchored by a protein called AMP kinase. AMP kinase is a positive regulator of autophagy. The remarkable thing about AMP kinase is that first, it can be activated by starvation, so it makes sense because I told you that starvation triggers autophagy, and this is actually part of the reason is because it went through the signaling pathway that is anchored around AMP kinase. Can we actually manipulate the autophagy process pharmacologically or using drugs? If autophagy process is so important, perhaps it's good for us, even though if we are not starving, sometimes maybe we want to trigger it. So can we do it in another way? It turned out actually AMP kinase can be activated by your garden variety painkiller, aspirin. So next time you take aspirin, think about autophagy. And actually also, a drug that is being commonly prescribed for type 2 diabetes called metformin is also a potent activator of AMP kinase. And so metformin actually is a drug that is very useful and is actually being given to a lot of diabetic patients. And yet the full spectrum of its beneficial effects may not be completely understood. But one beneficial effect perhaps is its ability to activate AMP kinase and all the downstream processes that are being controlled by AMP kinase. So finally, it's all nice and good. Now I hope that you would agree with me that autophagy is very important. But what happened? So to further convince you this is the case, I will tell you what happened when autophagy goes wrong, when there's no recycling. So being on this campus, I'm sure you get emails every other week talking about sustainability, that recycling is good, and recycling is good. Because if there's no recycling, things can go horribly wrong. So I'm going to give you 3 examples when there's no recycling, what happened. First of all, is neurodegeneration. So it turned out that actually our neurons needs energy because we think, you're listening, I'm talking. All these processes requires our neurons to work very hard. And neurons need energy to work very hard. So energy comes from healthy mitochondria. This is the powerhouse, it's the place where energy is being generated in every cell. But mitochondria can get overworked, and mitochondria can also get old, and the inside of the mitochondria get damaged. When they don't work very well, since mitochondria is so important that a cell needs to get rid of the bad ones, one of the ways to get rid of bad mitochondria is to actually put the bad stuff of the mitochondria on one side, keep the good ones, or keep the good parts of the mitochondria in another side, and actually split them. You can split a mitochondria, this process so-called mitochondrial fission, and so that the bad components, the bad contents of mitochondria are kept in one small bag, and you still retain the good parts that can work for your neurons. The bad ones now then can be targeted for recycling, and the recycling is done by autophagy. And so this is the conventional pathway of autophagy then. You're now degrading or recycling a complete organelle, but this organelle is damaged. This is the elimination of mitochondria, and it was given a special name, or mitophagy. And this also gave me an opportunity to kind of touch on another concept of autophagy, which is specificity. So how do the cells know or recognize the damaged mitochondria and spare the healthy ones? It turned out actually this would be a very universal concept of recycling. Autophagy process actually have ways to recognize the pattern. And just like in real life, if you see this Japanese recycling bin, you wouldn't be putting the wrong things in the wrong compartment because you know instantly that this would be for newspapers, even though if you don't read Chinese because this fits newspapers. And this would be for bottles, plastic or metal. Pattern recognition. So autophagy actually has a way of recognizing the patterns that was displayed on the surface of the bad mitochondria versus the ones that are healthy. And so in real life and inside our cells, the same idea holds true, that by recognizing specific patterns of damaged organelles, autophagy would not do the wrong thing, would not go for the wrong target. And so the idea is that then what happened when there's no recycling, when autophagy stopped working, then you get this progressive accumulation of the bad mitochondria, which is labeled in yellow here. And the end result is that all these defective mitochondria would actually make your neurons very unhappy. They don't get the energy that they need, and eventually would lead perhaps to the death of these neurons. Neurons are particularly sensitive to this process because, as I said earlier, whatever we do, we need our neurons to work. Our brain is working very hard, even as we sit and listen or talk. And so neurons need a lot of energy. And so if the powerhouse is broken, then the neurons would not be very happy, and so they might sometimes die. So the pattern of the broken mitochondria. So these patterns is actually come in the flavor of proteins. The patterns that was being recognized are actually two proteins. One is called PINK1, and the protein that recognized this damaged pattern is called parkin. These two proteins are famous, actually, not only for autophagy, but actually for a very well-known conditions of early onset Parkinson's disease. So it turns out that when PINK1 or parkin, these two important molecules that labeled damaged mitochondria and set them apart from healthy mitochondria, they would result in humans familial cases of Parkinson's disease. And what is horrific about this is that the age of onset is less than 40 years old, while normal, sporadic Parkinson's disease would occur in perhaps people aged around 60 or older. And so this would be a clear demonstration that recognition of damaged mitochondria and the process of eliminating damaged mitochondria by autophagy is extremely important for the health of neurons. Now, cancer. Autophagy turns out also important to curb cancer, to stop us from having cancer. And this is demonstrated by the study of yet another protein found initially actually in yeast, a protein called ATG16, in human called bacillin-1. We're back to the initial process of autophagosome formation. ATG16 or bacillin-1 is involved, again, being recruited in this very precursor form of autophagosome, the very beginning of autophagy. And this is absolutely essential for autophagy to occur. And what is remarkable is that, so first of all, just to have everyone on the same page, we all normally have 2 copies of the gene, of each gene. So we have normally 2 copies of the BACLin1 gene. The remarkable thing is, in fact, even if we lose 1 copy of BACLin1, so not complete elimination of the autophagy process, but just some retardation or just some impairment in the autophagy process, it is bad enough that losing 1 copy of BACLin1 may predispose us of having ovarian or breast cancers in a lot of the cases of these sporadic breast cancers and ovarian cancers. And so this argues that a reduction in the efficiency of autophagy is perhaps devastating in how we can suppress tumor formation. This was recapitulated in a mouse model, and so we know that this is a conserved process, that suppression of tumor by the autophagy pathway is not only in human, but actually in other organisms. And why only 1 copy? Why losing 1 copy of the BACLin1 gene? What happens if we lose all BACLin1 gene? It turns out if you lose both copies of BACLin1 gene, you probably would never be born. And so you don't actually see people who have lost 2 copies of BACLin1 gene and come and tell you, well, unfortunately, I've just got cancer. Because normally, if you lose both copies and you completely block the autophagic process, actually, you'll never be born. Even if you're born, you'll probably die within a day. And that happened in the mouse models. So again, so this argues that autophagy is an absolutely essential process for embryonic development, and even for later in life, any kind of reduction in the efficiency of autophagy would predispose individuals to terrible diseases. Okay, so turned out actually there's some more crosstalks between autophagy and known pathways of cancer. So it is well known, and the way is as follows. So there is a signaling pathway called epidermal growth factor signaling pathway, and this is well known to inactivate BACLin1 and inhibits autophagy. But you may have heard of EGF signaling in other contexts, because EGF signaling, the overactive EGF signaling, actually also promotes tumor progression. This is something that happened in many cases of lung cancers and breast cancers. And so the connection would be that perhaps part of the reason for EGF signaling to cause cancer progression or tumor progression would be the fact that it can also inhibit autophagy. And what is known, so pharmacologically, you can actually manipulate this process. There is actually a drug that actually can be prescribed to treat metastatic non-small cell lung cancer. This is a drug specifically inhibit the EGF signaling pathway. And in laboratories, people have been able to demonstrate that if you use this drug to treat cells, then the autophagy pathway would actually go back to normal in cells that are normally having overactive EGF signaling. And so that tells us that there's some biochemical connections in both directions between EGF signaling, autophagy, and cancer. And finally, this is the last example. I want to show you how autophagy is also important in metabolic diseases, or how when autophagy goes wrong, it can lead to metabolic disturbances. So this is more like a self-advertisement, because actually up till now you may wonder, why am I giving this presentation? And why am I in a position to tell you about autophagy? So my lab actually studies how fat is being stored and packaged inside a cell. And we're interested in an organelle called lipid droplets. This is the place where fat is actually being, again, kept in nice little plastic bags inside a cell, where either fat from your diet, from your lunch, or fats that we made in our body are kept nice and tight inside a cell, forming these spherical packages. So all the lipid droplets are labeled in red in these 2 cells. So how do you actually get fat out of the lipid droplets? Because it's not so useful if you can only store them. I mean, this is actually store them for emergency, right? Store them when you need them, but you want to actually be able to withdraw the fat when you need them. Again, for energy. Right? Fat is a very important precursors that would give us a lot of energy. It turns out there are two ways that you can actually get fat out of lipid droplets, either by enzymatic breakdown of the fat that is stored inside the lipid droplets, or you can actually get rid of lipid droplets, the entire organelle, by autophagy. And this is yet another specialized form of autophagy, and this is given the name lipophagy. So what's the difference between these two processes? How do you decide which process to use? So the enzymatic breakdown would be like taking cash out of an ATM machine. The ATM machine has a lot of cash. All the cash is your fat inside lipid droplets. You are taking out some cash a little bit at a time. That's enzymatic breakdown. What happened to lipophagy? Lipophagy would be at the time when you're quite desperate. Actually, you don't even have time to get cash out of the cash machine. You just take the entire cash machine away. That would be lipophagy. You're taking everything, all the contents from lipid droplets, and you say, I want everything. And so then you take your lipid droplets, deliver them to the lysosomes, and you completely strip all the fat out of lipid droplets. What happens when this goes wrong? Or where does it happen? In what tissue, in what organs would the cell be so desperate to get the entire ATM machine? It's actually more common than you think. Actually, between these two ways of getting fat out of lipid droplets, in the liver, in our livers, in mammalian livers, actually, the ATM machine removal, lipophagy, is actually the preferred way of getting fat out of lipid droplets. So how can we demonstrate this? So in fact, that if you block lipophagy in liver, you'll end up with fatty liver, or foie gras. So this, again, is not something you want. I mean, fatty liver disease is a devastating disease, a serious metabolic problem. And so that, again argues that autophagy is important in this particular case to control the level of fat that we should maintain in the liver. All right, so this is the summary. So autophagy has been getting more and more popular. This is the cumulative number of papers that you can find in PubMed. And what is remarkable is that this entire field, and what justifies, perhaps, to the award of Yoshinori Osumi for his work in autophagy, is that you can see that the reason of this rapid rise in the autophagy research is really can be contributed to his pioneering work. First, he demonstrated that autophagy can occur in yeast. He isolated yeast autophagy mutants using genetics. He demonstrated the gene products he found. What are these ADG genes and what do they encode? He makes the field much more popular by demonstrating that the yeast autophagy pathway is actually conserved in mammalian systems. And finally, he demonstrated the biochemical reactions that was important in this autophagy process. And as a result, also demonstrated the use of a very useful marker to monitor autophagy. And really it's really after 2000, then you can immediately see that the laydown of the signaling pathways and the creation of tools that allow other people to study autophagy in not just yeast, but also in other systems that allow this field to really explode in the last decade. So just to summarize, that autophagy is important, that is required for embryogenesis, for just developing from the very, very early stages of our life. It's important to counter starvation and other types of stress because it allows us to recycle redundant, unwanted materials to give something useful for our cells to use. Without autophagy, it may cause neurodegenerative conditions, predispose us to cancer, and also make us more prone to bacterial or viral infections. So I hope I've convinced you that this year's Nobel Prize is the winner, is a worthy winner, and that don't get too starved. Because actually, well, we'll be fed, I guess, after this, right? So our level of starvation will be rather limited. So thank you very much for coming.
Thank you for a great talk. So you described 3 examples of disease conditions where you have, say, no autophagy or defective autophagy. So I'm curious, is there any disease conditions where, say it's a result of overactivation of autophagy?
Yeah, so the question is whether overactive autophagy would be bad for you. I'm not aware of it, actually. So one thing I want to also, maybe I kind of skimmed over, is actually in all of our cells, actually we have a low constant level of autophagy that is happening all the time, whether we're starved or not. That's called basal autophagy. And then when we are starved, or when we have a lot of damaged material, then there will be an induction of extra autophagy. And so I think in our cells, we don't mind to have autophagy going on all the time, albeit at low level. So maybe, but you give me an idea to go back and actually find any bad diseases for having overactive autophagy. So I can't really answer your question at this point.
Very interesting talk today. So I actually have 2 questions about the liposomes here. So the liposome is very important for the autophagy event. So where does this lysosome originated? Is it derived from other organelle, or can it undergo some fission in fan?
Yeah, so I think you're referring to, so you're asking basically the origin of kind of the precursor form of autophagosomes. So this turned out to be quite controversial. Lots of people were still studying this process. Some suggested that these membranes came from the endoplasmic reticulum, for example. It's not so surprising because endoplasmic reticulum is quite extensive in any cells. Some would argue that it may also have additional origin. And so right now there is no conclusive answer to, or there's no consensus as to where these membranes are all coming from, but one suspect, or one source, one potential source would be the endoplasmic reticulum.
And the second question would be, so since the lysosome is responsible for scavenging the kind of the wrong organelles, or the organelles undergo some bad processes. So what if the lysosome itself get wrong? So what could be the things that will scavenge the wrong lysosome?
Yeah, that's a great question. So I forgot to mention that actually your lysosome, as you said, lysosomes can also stop working. Your recycling factory can stop working. And actually many of the genetic diseases that are involved in lysosomes being not able to work is also neurodegeneration. So there's actually a large number of genetic diseases in which enzymes that normally work in lysosomes, when they are defective, then they will, and children in those families would end up actually having severe neurological problems because the neurons don't function very well. So that's actually quite similar to the conditions where autophagy doesn't work. Right? So it's actually the same process. It's actually you're stopping the same process at different points.
Let's thank Professor Mack again.